everyone, welcome back to my YouTube channel. In this video we will talk about neutrophil extracellular traps, which are in shortcut will known as NETs. Our extracellular fibular network are concentrate an antimicrobial substance at sites of the infection and prevent the spread of the microbes by trapping them in the fibrils. They are produced by the neutrophil in response to the infectious pathogens and inflammatory mediators. The extracellular traps consist of a viscous meshwork of nuclear chromatin that bind and concentrate granule proteins such as antimicrobial peptide and enzyme. NETs provide an additional mechanism of killing microbes that does not involve the phagocytosis. In the process of the NET formation, nuclei of the neutrophil are lost, leading to the death of the cells, sometimes called NETosis, representing a distinctive form of the cell death affecting the neutrophil. NETs also have been detected in the blood during the sepsis. The nuclear chromatin in the NETs, which include histones and associated DNA, may be a source of nuclear antigen in the systemic autoimmune disease, particularly lupus, in which the individual react against their own DNA and nucleoproteins. Leukocyte-mediated tissue injury. Leukocytes are important mediator of the injury to normal cells and tissue under the several circumstances. As a part of the normal defense reaction against infection, microbes when the tissue at or near the site of the infection suffer collateral damage. In some infections that are difficult to eradicate, such as tuberculosis and certain viral diseases such as hepatitis, and prolonged host response contribute more to the pathology than does the microbe itself. When inflammatory response is inappropriately directed against the host tissue, as in certain autoimmune diseases, when the host hyperreacts against usually harmless environment substance, as in allergic diseases including asthma and some drug reactions, leukocyte damage tissue by releasing injurious molecules. The potentially toxic content of granules are released by the leukocytes into the extracellular melee by the several mechanisms. Controlled secretion of the granule content following degranulation is a normal response of activated leukocytes. If phagocytes encounter materials that cannot be easily ingested, such as immune complexes, deposit or immovable flat surfaces, the initially of the leukocyte to the surround and ingest these substances. Frustrated phagocytosis triggers strong activation and also release of the large amount of the granule enzyme into the extracellular environment. Some phagocytized substances such as urate and silica crystal may damage membrane of the phagocytosome and others lead to the release of the damaging content. The neutrophil can exclude the nuclear content from extracellular nets and are trap and destroy the microbes. Granule enzymes may be released into the extracellular environment, the mechanism that functions to eliminate the microbes and dead cells. Physiological role of the inflammation are capable of damaging the normal tissue. The pathological consequences of inflammation. Anti-inflammatory mediator terminate the acute inflammatory reaction when it is no longer needed. Other functional response of the activated leukocytes. In addition to eliminating the microbes and dead cells, activated leukocytes play several other roles through host defense. Importantly, these cells, especially macrophages, produce cytokines that can either amplify or limit the inflammatory reactions. Growth factors that stimulate the proliferation of endothelial cells and fibroblasts and the synthesis of the collagen and enzyme that remodel connective tissue because of these activities. Macrophages also have central roles in outstretching chronic inflammation, and these repair after the inflammation has subsides. These functions and macrophages are discussed later in the chapter. In this discussion of the acute inflammation, we emphasize the importance of the neutrophil and macrophages. However, it has become clear that some T lymphocytes, which are cells of adaptive immunity, also contribute to acute inflammations of adaptive immunity. The most important of these cells are those that produce the cytokine interleukin 17, which are discussed in more detail in next upcoming videos. Thank you so much for watching the video. Keep watching, keep supporting.